but how, where, and when it happened. In Africa, Raymond Dart finally finds a kindred spirit, an eccentric physician and zoologist turned fossil hunter, Robert Broom. On his first visit to Dart, Broom kneels before the Tong skull to pay homage. Widely recognized for his work classifying mammals in South Africa, Broom likes to flaunt authority. He has already tarnished his reputation by selling fossils abroad. Determined to validate Dart's find, he sets out in search of an adult to match the Tong child, now known as Australopithecus africanus. Guided by Dart's students, Broom visits the Sterkfontein lime deposits near his home. Within days, he finds fossil fragments and shows them off during a lecture.
By the way, he would say after 2 hours of lecturing, and out would come what he described as 2 of the most marvelous teeth that have ever been discovered in the history of the world. And these were Australopithecus teeth.
After 10 more years of persistent searching, the 80-year-old Broom finally finds a complete skull. It had a small, ape-like brain, but it walked like a human being. Dart's Tong skull is no longer an isolated find.
It came to be realized that there was not one freakish little skull, but that there was a whole community, a population. It was the accumulation of new evidence that turned the tide.
Then in London in November 1953, one of the greatest scientific hoaxes of the 20th century is uncovered.
Britain's august Natural History Museum is all a-dither over a scandal concerning the Piltdown Man. One of the most famous fossil skulls in the world is declared to be in part a hoax. 40 years ago, its discovery was a sensation. Today comes the shocking news that this is skullduggery.
Piltdown was guarded, I think, even more closely than the crown jewels. There wasn't an opportunity to really study the specimen in depth until the early 1950s, when the Piltdown was on display. A fellow by the name of Viner had an opportunity to see it. And he thought that there was something wrong here, that this specimen didn't really fit together.
When Kenneth Oakley, curator of the British Museum, uses fluorine dating, he discovers the skull is 10,000 years older than the jawbone.
A devastating publication came out and showed that this was, in fact, the jaw, the lower jaw of an orangutan that had been doctored to look like a hominid jaw. We don't know who actually perpetrated this or for what reason. Some people think it was to embarrass other English scientists into believing that this really was a bona fide human ancestor.
With Piltdown discredited, the Tong child is now embraced as our rightful ancestor. Africa appears to be the cradle of humanity after all. Over the following years, its terrain offers ever more clues to the mysteries of human origins.
It's because of the geology that we have such good fossil remains in places like Eastern and Southern Africa. The development of the Great Rift Valley, for example, that's been going on for tens of millions of years, has formed a series of collection places. Lakes and basins and rivers for animals to fall into and become fossilized.
In 1972, Richard Leakey, following in the footsteps of his mother and father, adds to the growing trove of African fossils with the discovery of Homo habilis, almost 2 million years old. Then, in 1974, Don Johansson's research team sets out for East Africa.
I was co-leading an expedition to the Afar Triangle, and I had just finished my PhD at the University of Chicago that year, and my great hope was to make a major discovery, something important that would help us fill in this picture of our earliest origins. I was out surveying. I happened to glance over my shoulder and spotted a piece of elbow and recognized it immediately as coming from a human ancestor. This wasn't a baboon or an antelope or any other kind of animal. It had to come from us. And as I kneeled down to pick it up and look at it more closely, I saw other pieces. I saw pieces of, for example, here is a leg bone. The black portions are reconstructed parts, but we had most of the femur or thigh bone. And as we continued to collect fragments from this hillside and piece them together, we came to realize that we had most of a single skeleton. And there were some astonishing discoveries. The length of her legs, for example, suggested that she was only about 3.5 feet tall, much shorter than we are today. The immediate question was, are we dealing with a child, like Dart was with the Tong baby? Well, when you look at the teeth, you see that the permanent molars, the adult molars, have erupted. So this was an adult creature. And because of its small size, we surmised that it was a female.
They call her Lucy, after the Beatles song they played the night they found her. When she lived, she was about the size of a chimpanzee. And like Dart and Broom's finds, she walked upright and had a small brain.
We think Lucy's skull might have looked something like this, with a receding forehead and a prominent face. And with a brain case no larger than a chimp's, this was no smart ape.
In 1974, Lucy is the most complete hominid fossil yet found. She is more than 3 million years old. But the path through those years, from Lucy to us, was, like the rest of evolution, far from straight.
As we look back and explore and discover fragments of our ancestry, we see that it wasn't a direct line from Dart's Tong child or from Lucy or whatever directly to us. That there were many false starts, that there were branches, there were extinctions. Every single one of these fossils we find is a link. It's a link to our past, but even more importantly, it's a link to the natural world. And this is a tremendous blow to people who think of themselves as the pinnacle of evolution, the chosen species. That we are not necessarily here because of some preordained idea, that we have gotten here through the same process as all other life.
Twentieth century paleoanthropologists got more than they bargained for when they tackled the mysteries of human evolution. But it was even harder for science to uncover the fundamental rules that govern the evolution of life itself. How could ancient animals and plants like these fossilized ones evolve into the incredible variety of species we see today? What mechanism could explain an individual's characteristics being passed down to future generations? How could an entirely new species be created? The first breakthrough occurred in 1900 when the work of an obscure Augustinian monk named Gregor Mendel was rediscovered 35 years after his death. Mendel demonstrated that the physical characteristics of pea plants, like this one, were inherited according to certain rules. Eventually, he could even predict which offspring would be short or tall, darker or lighter in color, depending on the characteristics of previous generations. But Mendel had no idea how these traits were carried from one generation to another. What told a pea to be wrinkled or smooth? For Darwin's theory of evolution to be fully accepted, someone had to find the mysterious something that makes it all work. According to Darwin, species evolve through a process called natural selection. When traits, like the size of a fin, are passed down from one generation to the next, some random variation or mutation may occur. When this mutation helps an organism adapt to its environment, the organism is more likely to survive and pass the altered trait on to its offspring. It's through this process that a species changes or evolves. And if the accumulated variations are large enough, a whole new species can arise. As the 20th century dawns, many scientists find it hard to accept the randomness of natural selection. And no one understands how mutations actually work, or how traits are passed down through the generations. In 1904, a biologist named Thomas Hunt Morgan begins a series of experiments he hopes will uncover evolution's secrets.
Morgan believed that modern-day species had descended with modification from previously existing species. So in that sense, he was very much alongside of Darwin. What he had trouble with, as did many of his contemporaries, was the question of how you could get two species by selecting small, minute variations from one original parental population. The experience of practical animal and plant breeders, like dog breeders or horse breeders, which he was familiar with from Kentucky and so on, suggested that though you could make many improvements in these organisms by selection, no one had ever produced a new species that way.
Morgan grew up in Kentucky right after the Civil War. His uncle was Brigadier General John Hunt Morgan, called the Thunderbolt of the Confederacy, who had commanded a daring band of guerrilla soldiers known as Morgan's Raiders. As a boy, Morgan felt a strong attraction to the countryside around Lexington. He saw the great swaths cut into the hills to prepare for the coming railroads, and was fascinated by the exposed layers of sedimentary rock and the abundance of fossils.
My grandfather was always a little different. He liked to go out collecting things in the woods, stones, feathers, fossils, skeletons of animals, and he would collect things and have his own little world.
Morgan's love of nature leads to a PhD in biology. Then, in 1903, while teaching at Bryn Mawr College, Morgan hears exciting news. Hugo de Vries, a Dutch biologist, discovers what appears to be a new species of plant, growing right alongside its parent, originally thought to be produced by a single large mutation.
When Morgan read de Vries's work, he felt that might be the answer to the problem that he didn't think Darwin had solved, which is how you get from one species to a second, very different species. And so he became very interested in de Vries's work, but de Vries had worked with plants. Morgan was a zoologist, and he decided he was going to try to generate and find those kind of mutations in animals.
Morgan chooses the common fruit fly, known as Drosophila, for his experiments. It's a perfect choice.
A female fly will produce 200-300 progeny easily. Secondly, they're easy to raise. You can raise them on a simple medium. You could grow them on mashed bananas. A third, you could separate males from females easily. The life cycle was short, so that in a matter of 10, 12, 13 days, depending upon the temperature, you had a generation of flies. So we started out just looking for mutations.
The work of sorting through thousands of tiny insects in search of mutations is extremely laborious. And it seems futile.
For several years, he didn't get any fruit flies that looked substantially different from their parents, and he was just about to give up the experiment. In fact, he wrote to a colleague in Europe and said, you know, I've just about had it with these fruit flies. They're not producing anything. It's been several years of work, and I'm just going to abandon it and go on to something else. And just shortly after that, he discovered in his culture one white-eyed male fruit fly.
Finally, a mutation. Not a new species, but Morgan is intrigued. Instead of looking for large-scale, de Vries-like mutations that might drive evolution, he decides to investigate small variations, like the white eye. He breeds the white-eyed male to a normal red-eyed female. Will the mutation be passed on to the next generation, making the offspring white-eyed? Or will they have normal red eyes? Or even show a blend of red and white and have pink eyes?
There was no consensus about how inheritance worked. No one really knew how traits were passed on, whether there were any patterns or laws. It was clear that like begets like, that offspring look somewhat like their parents, but sometimes they look like their grandparents, or sometimes their great-grandparents, or sometimes like no one in the family. And there were about as many theories, as one author said, of heredity as there were people proposing them.
The work of Gregor Mendel, however, stands out. Mendel had found that when he bred tall plants with short, the first generation, the F1s, all turned out tall. But some short plants reappeared in the next generation, in a ratio of about 3 tall to 1 short. Mendel concluded that among those 3/4 that were tall, 2 contained both tall and short characteristics, but the tall was masking the short. Mendel believed that all the plant's characteristics were controlled by some factor from each parent that was passed on to the next generation, and that these factors, later called genes, never blended together to form new ones. They remained discrete, determining the characteristics of offspring by their different combinations.
Although Mendel laid out genetics very clearly in 1865, it really didn't catch on. I think the paper was largely ignored for the next 35 years. I think the reason is that although this abstract concept of factors or genes controlling inheritance was an interesting one, hard-headed biologists would say, well, where are the genes? Can you show them to me? There was nothing physical to see about genes.
Morgan is aware of Mendel's work, but remains skeptical.
He asked a very simple question. What are these things that Mendel called factors? How are they transmitted from one generation to the next? What's the basis for all of this? Are these units? Are they single? Are there multiples? What's going on here?
By 1910, Morgan and his wife Lillian, also a notable biologist, have produced a new generation of Morgans. They call them their F1s. They spend their summers in Woods Hole, Massachusetts, where Morgan builds a big house half a mile from the ocean. The Morgans will summer at Woods Hole for the next 40 years.
Every summer they would pack up these milk bottles full of fruit flies and bring them up to the Marine Biological Laboratory in Woods Hole and work with them and do their breeding and do their cataloging experiments here.
We loved this place. We'd run off down to the eel pond and see what was going on there. We'd go out collecting in tide pools and make slides and study. It was a very close and informal community. The scientists that my grandfather knew were always in and out of the house.
Morgan would take his group out on the lawn outside of one of the laboratories at the Marine Biological Lab, and they would simply lie on the grass and talk about their day's results or the week's breeding figures and so on.
Back in New York City, Morgan's breeding experiments with his white-eyed fly begin to show the kind of results Mendel's theories predict.
He bred this white-eyed fly to a red-eyed fly and looked at the offspring. And all the offspring were red-eyed. But then he asked, being familiar with Mendel's work, he thought, well, now Mendel observed that with his peas, that all the offspring of the first cross looked alike. So what happens if you cross those with each other? He did that, and lo and behold, he got about 25% white-eyed. But then he noticed something that was really peculiar. Every one of those flies was a male.
How come only males? Why not females? Then he did a little thinking, and he said, what in a fly, or what in any animal, starting from the grandfather, will end up in a grandson? What's the only thing?
A clue lay in a discovery of the late 19th century, when biologists had seen pairs of long, thread-like structures in the nuclei of cells. They called them chromosomes, after the Greek word for color, since these structures absorbed dye readily and stood out clearly under the microscope. They found that a particular configuration of chromosomes seems to have a role in determining sex. And since the white eye only shows up in males, Morgan suggests that perhaps a Mendelian factor for eye color is associated with the chromosome configuration that determines maleness.
Morgan did not go as far as to say that this factor for eye color was actually physically a part of the chromosome, just that it always appeared with it in the offspring. Within a year, Morgan had found several other characteristics that also seemed to be sex-linked. And at that point, he was willing to say that the factors determining these particular traits were actually physically part of the chromosome itself.
So he published a paper, and he postulated that the white eye gene, if there is such a thing, must be on the chromosome. That was the big step.
Morgan continues searching for more traits that are linked to convince himself that factors of inheritance, genes, are physically located on chromosomes. It is tedious, exacting work that can only be done with a devoted team of researchers.
In the Morgan lab, there was a congeniality, a give and take. It generated the kind of thinking which was necessary to do these crucial experiments. And Morgan let people, gave him free license.
Very few research projects at that time were done in groups, and Morgan's group of young undergraduates and then later graduate students became the prototype of a whole new way of doing science. compared to the European tradition where the professor in charge of a lab was very austere, very standoffish, would maybe come through the lab once a day and talk to a few people about what they were doing. Morgan was just the opposite. He sat at his own desk in the fly room. He had his own microscope. He would count flies along with everybody else. There was a tremendous amount of informality.
Morgan and his team study over 13 million flies. Gradually, during the early 1920s, a genetic map of Drosophila starts to emerge. They begin to understand where on the chromosome genes for particular traits are located.
And they became convinced now that you were dealing with a material particle that was indeed passed on from generation to generation during the period of cell division. This gave them a sense that now they were gaining an understanding of how heredity could occur in material fashion and in such fashion that you could alter it in the laboratory and predict the outcomes, something that was very, very difficult to do with evolution itself.
Morgan's work convinces him that evolutionary change does not require large-scale mutations. He has observed so many small variations in Drosophila over the years that he comes to believe, like Darwin, that small, incremental change is enough to propel evolution.
Morgan gave rise in his lab at Columbia to a whole school of geneticists that laid down all of the fundamental rules of heredity that shaped the rest of the century, including all the fundamental rules we apply today in human genetics.
But what the genetic material was, how it was organized in the chromosome, precisely how the chromosomes replicated and how you replicated the genetic material, and what it actually does in the developmental process, that was a black box.
Scientists knew that what was in the black box could in some way be called the secret of life. The beauty and wonder of life itself. The incredible diversity that had puzzled and intrigued Darwin and Morgan. Could this be reduced to chemistry? Understanding the chemical basis of life had become the ultimate challenge for many scientists.
The fascination was, can you put together the ordinary atoms that chemists know, carbon, phosphorus, hydrogen, and others, and make something that's alive? What is the physical basis by which something can reproduce itself? How does chemistry and physics explain that? Maybe there's something new, a new principle that we don't know. Is it like that Michelangelo is a spark that has to go from one finger to another? Or what is it that makes something alive?
To be alive and grow, the cells in an organism must be able to reproduce and pass along their genetic information to the next generation. Whether it's a human embryo or a blade of grass. Morgan showed that genes transmit information from parents to offspring. But how? By the 1940s, scientists know that genes contain DNA, short for deoxyribonucleic acid. Either proteins, which do most of the work of the cell, or DNA must contain the secret of heredity. But which one?
All smart, well, right-thinking people at that time knew that DNA was an utterly boring structural molecule that couldn't possibly contain anything as interesting as heredity. Proteins had to be the stuff that contained heredity because proteins were very variable and very interesting. And yet the experiments kept pointing to the fact that it was DNA which, when transferred to a new organism, contained the secret of heredity.
In 1951, at King's College in London, researcher Rosalind Franklin takes X-ray photographs of DNA molecules. At 33, her scientific accomplishment is testament to her determination. She defied her father, a London banker who thought education was unhealthy for women and had wanted her to be a social worker. Instead, Franklin has become an expert X-ray crystallographer. And her latest photograph of DNA suggests a helical or spiral shape. Cautious by nature, she decides she needs more evidence before sharing her findings. She tucks the photo into a drawer. "60 miles away at Cambridge University, James Watson, a brash 24-year-old American geneticist, is also consumed by the study of DNA. As a boy, he starred on The Quiz Kids, a Chicago radio show, and by age 22, he'd earned his PhD. Working with Watson is British physicist Francis Crick, of whom Watson will write, "I have never seen Francis Crick in a modest mood. He talked louder and faster than anyone else." Crick had studied physics at University College London, but at 35 has yet to complete his doctorate. As they recall 20 years later, somehow it's a match made in heaven.
He thought like I did. Neither of us had sort of preconceptions. Neither of us were trained for what really interested us now.
As you said, we had the same point of view. And of course, I found the same reaction to you as you found to me. I was self-taught. I was stuck in Cambridge. There weren't that number of people who I could communicate with.
Later that year, on a trip to London, Watson is shown the X-ray Franklin is not ready to share publicly. "My heart began to race," he says later. He rushes back to Cambridge to tell Crick that the shape of the DNA molecule may well be a helix.
They then began the task of literally constructing a model. Back in Cambridge, at the Cavendish, they took pieces and they tried putting them together in different ways.
Certainly they were working at a fever pitch.
Was it a race? Of course it was a race. Who wants to discover one of the secrets of the universe second? And they went after it with full force.
They come up with a model that looks promising.
I was very excited, probably because Francis was excited. It was our first DNA model.
It did look like a sort of cylinder of things, didn't it, Jim?
Sort of a solid cylinder.
With things sticking out, roughly speaking.
It wasn't very pretty.
We were completely on the wrong track. So when you have an idea and you've been enthusiastic about it, and it's clear it's wrong, you must just push it away and get rid of it.
DNA has 4 chemical components, or bases, called adenine, thymine, cytosine, and guanine, or A, T, C, and G. The bases seem to occur in pairs. So Watson decides to try something else.
I cut some things out of cardboard and sort of made the right shapes and then pasted things on which would indicate hydrogen atom. Then I think I went off and played tennis. And it wasn't until the following morning I came in and I started to put them together in pairs.
When Watson makes models of the base molecules, he realizes that when they're combined, A with T and G with C, each pair has the same overall shape.
It all fell together. And when it fell together, it was so obviously right that they had to have known, they did know, that they had cracked one of the major secrets of the universe.
Watson and Crick suddenly realized that the structure of DNA is a double helix. with the AT and GC bases on the inside of the molecule, shown here as red and green, yellow and blue. The base pairs connect to each other between two ladder-like chains of sugars on the outside, all of it twisted like a double corkscrew. Watson and Crick published their findings in Nature in May 1953, sending shockwaves throughout the scientific world.
I was in the Department of Chemistry at Caltech, and Max Delbruck was in the Department of Biology. He asked, "What do you think of the paper by Watson and Crick?" I said, "I'd never heard of these papers." So he reached behind to a stack of reprints he had on a shelf behind his desk, and he threw the whole heap of reprints, because Jim Watson had sent him a bunch of reprints of the two papers, at me. And there this floor is covered with these little reprints. And he said, "Read it carefully. The most important discovery in biology in 10 years. Later, I wondered why he didn't make it 50 years. In 10 years, and you don't know about it.
In 1962, the Nobel Prize goes to Watson and Crick for discovering the structure of the DNA molecule. But Rosalind Franklin does not share it. She had died of cancer several years earlier at the age of 37. With the discovery of the structure of DNA comes an understanding of how chemical molecules can pass on the code of heredity.
The real significance of the discovery of the structure of the DNA molecule was that heredity, which you could have imagined to be infinitely complicated, was in fact very simple.
DNA's double helix can unzip to make copies of itself. Each half becomes a template that attracts matching bases to complete a new molecule.
And this was the great secret. Any one strand, because it was a perfect match for the other strand, contained all the information. You could take away one of the two strands of the double helix, and you'd still have all the information left on the other. Why was that the secret of life? Because it explained how organisms replicate, how they pass on genes.
Normally, the copy is exact, and parental characteristics are passed on. But with billions of pairs to duplicate, the occasional error is inevitable. The result is a misspelling or mutation, like Morgan's white-eyed fruit fly.
It's a slow and steady accretion of misspellings, maybe at a rate of only one letter in a million. But then selected upon by natural selection. It gives rise to all the variations that change the simplest plant cell to a sequoia tree. And that is the basis of all the diversity on the planet Earth.
This extracted DNA is a virtual window on evolution, a path to our past and to the past of all living things.
DNA change is a clock mechanism that allows us to date things, to date when two species had a common ancestor, when different branches of evolution occurred.
Our DNA is only 1% different from that of a chimpanzee. In fact, we're closer to chimps than chimps are to apes. It is now believed that humans and chimpanzees diverged from a common ancestor only 5 to 8 million years ago. Through DNA analysis, we can even trace our ancestry back 2 billion years to bacteria and yeasts. Human DNA and yeast DNA are still so similar that human DNA can repair yeast cells which can no longer divide. When human DNA is implanted in them, the defective yeast cells begin dividing again.
That tells us better than any fossil record ever could that we must have a common evolutionary ancestor, a common great-great-great-great-great-great-great-grandparent with a yeast cell that goes back all those billions of years. A measly 3 billion letters of DNA, just the amount of information you could put on a single CD-ROM, contains enough to specify all of the remarkable diversity within a human body and amongst humans. The structure of the DNA molecule explained everything. It laid bare the secrets of life, of reproduction, of mutation, of natural selection. It shows us that the fundamental processes underlying all of life are the same in all living organisms. It is an absolutely stunning unity of life.
In the latter half of the 20th century, we come to understand that the arrangement of molecules in strands of DNA provides the information that tells cells what to do. In a person's hand or in a turtle's shell. That's a discovery that would have astounded biologists only a few decades earlier. But there's still the question of how life started in the first place. The Bible suggests that life sprang fully formed from the hands of God. The ancient Greeks, on the other hand, believed that when the earth was new, its surface fermented until parts of it swelled up. Then thin membranes formed around the swellings. And when the membranes broke, all kinds of animals were born. For centuries, scientists had few ideas of their own. But that begins to change in the 1950s. About the same time Watson and Crick are working on DNA, others begin to explore the mystery of life's origins. Many theories are proposed, but none is proved conclusively. And yet, as the century winds to a close, scientists are beginning to believe that we are only a few steps away from learning how it all began. The early Earth was hardly a suitable place for life. It was violent, incredibly hot from the constant bombardment of meteors. But in time, the meteors were less frequent. The Earth cooled, oceans formed, and somehow, somewhere, life managed to gain a foothold. Probably around 3.5 to 4 billion years ago, the first step from non-life to life was taken.
So what's life, and how did life ever get started? We have a pretty good understanding of how single-celled organisms with DNA, with particular genes, could evolve by slow mutation into complex organisms today. But where did those simple single-celled organisms come from?
What could have made atoms and molecules on the primordial Earth combine in just the right way to make life? In the 19th century, Charles Darwin suggested that the answer might lie in what he called a warm little pond.
He talked about a notion relatively common at the time, and that is life began in that warm little pond where you had a mix of chemicals, where you had the coursing through it of either strong blasts of heat or electricity, something like that, he said, out of which you would have the formation of some proto-form of life.
In 1953, Stanley Miller, a graduate student in chemistry, plans an experiment to see if the molecules necessary for life could have formed under conditions present on the early Earth. His advisor, Nobel Prize-winning chemist Harold Urey, believes that instead of being rich in oxygen, the atmosphere of the primordial Earth was more like the outer planets of the solar system. Rich in hydrogen, ammonia, and methane gas. But could life have really begun under these conditions?
This was a first-class scientific problem. I mean, I don't see how one can think about anything without asking where we came from, where life came from, and questions of that sort.
Miller and Urey build an apparatus to model the primitive Earth. A system of glass tubes and flasks in which water and atmospheric gases can circulate, subjected to jolts of electrical energy.
The water is like the ocean. The spark is somewhat like lightning. And the gas flask of about 5 liters corresponds to the gases in the atmosphere.
After about 24 hours, the once clear water becomes yellow. After a week, it's nearly brown. Miller's analysis of the liquid reveals that amino acids have formed where none had existed before.
The amount of amino acids produced was far larger than we even hoped we could get. Where we were confident that we could get lots of organic compounds, but that we should get one so closely connected to biology was the big surprise of the experiment.
Amino acids make up proteins, which, along with DNA, make life possible.
What Miller's experiment showed us was at least some of the basic building blocks clearly could arise on their own by chance. And it led to an exploration of what else could arise by chance. And how could the things that were easiest to arise by chance give rise to more things and more things? And how could you get a system that could sort of bootstrap itself up going? And so Miller's experiments didn't show us exactly how life arose, but it pointed the way to how you could begin to tease apart that problem.
And yet, other scientists reject the experiment's most basic assumption. What if Miller and Urey are wrong about the conditions on Earth 4 billion years ago?
In the 50s, it was thought that the early Earth had an atmosphere rich in methane and ammonia. In that kind of atmosphere, it's very easy to make organic molecules, and it's easy to see how to get the ball rolling for the origin of life. But the consensus opinion right now is that the early atmosphere was probably carbon dioxide rich, not methane rich. And in that kind of atmosphere, it's much, much, much harder to make organics. And that begins to push you to look at other possibilities.
In 1977, at Woods Hole, Massachusetts, another possibility for the origins of life is uncovered, almost accidentally. A miniature submarine, nicknamed Alvin, sets out to explore an area of the Atlantic Ocean where new molten seafloor is pushing apart the Earth's tectonic plates. Alvin's crew discovers large plumes of superheated water streaming from the ocean floor at nearly 700 degrees Fahrenheit. Volcano-like vents giving off black smoke. And most amazing of all, strange forms of life never before seen.
There are whole communities of organisms ranging from bacteria to crabs to long tube worms that make their living out of harvesting the chemical energy from these vents.
It used to be thought that the best source of energy at the beginning might be the sun. But we now understand that there are other sources of energy. It's possible that the Earth's own heat might have been the source of energy used by early life. Whatever it was, early life needed some reliable source of energy, a reliable source of chemicals, and a way to capture both of those efficiently.
Yet another theory suggests that the precursors of life didn't originate on Earth at all. This theory gains credibility when a meteorite is recovered in Australia in the fall of 1969.
Space scientists have discovered it contains amino acids, the building blocks of life, and found them in combinations that seem to prove they were there before this tiny piece of asteroid hit the Earth.
So there's this possibility that the building blocks of life on Earth didn't come from the Earth itself, but were imported. The meteorites that actually would be big enough to see, if you could look at them, they burn up in the atmosphere. But if you're smaller than that, if you're microscopic in size, you can enter the Earth's atmosphere and just get gently slowed down and not get burned to a crisp. Organic molecules in those dust particles, those microscopic dust particles, can make it intact to the surface of the Earth.
Could life have begun simply because a comet or meteoroid happened to approach the Earth in just the right way, at just the right time? The evolution of life is propelled by random events, so why not life's origins? In the late 1970s, a father and son team of American scientists proposes that another random meteor impact might be responsible for the massive destruction of life, a chance event that also shaped the course of evolution.
In the late 70s, Walter and Louis Alvarez were doing things that geologists love to do. They were simply measuring the rate of accumulation of material, and this was out in Italy, in Gubbio, Italy. They were measuring the rate of deposition, and they discovered something very unusual. They discovered a layer, a very thin zone, that had an enormous amount of iridium. Now, iridium is not a naturally occurring element. It's usually brought to Earth by the outside. It's brought to us by asteroids and comets colliding with the Earth.
The traces of iridium the Alvarez team finds in Italy come from rock strata 65 million years old. Soon, other 65 million-year-old iridium layers are found in other parts of the world, like here in southern Colorado. This date happens to coincide with the time in Earth's history when the dinosaurs went extinct after thriving for more than 150 million years.
This is the KT boundary. Below this horizon is the Cretaceous period, the time of the dinosaurs. Above this white layer, they were gone. The dinosaurs were simply gone.
The Alvarezes suggest that the impact of a giant meteor caused the dinosaurs' demise. It's a daring idea with enormous implications for our own origins.
If the dinosaurs had not gone extinct, we probably would not be here. Dinosaurs were enormously successful organisms, and if they hadn't been snuffed out, the mammals may never have risen to be the dominant form of life on the planet, and human beings might not be here. So in a very immediate sense, our origins might be tied up with big impacts.
Earlier in the century, most scientists did not believe that the Earth had ever been struck by large meteors. Even craters like this one in Arizona, now known as Meteor Crater, were thought by geologists to be made by volcanic action.
There was no consensus on whether the craters were volcanic or actually derived from impact. When the tide shifted in the late 50s and 60s toward the belief that indeed almost all of these features were a result of meteorite impact, it brought a phenomenal new understanding to the activity in the solar system.
Manned exploration of the moon in 1969 confirmed that moon craters were not the result of volcanoes, which had been widely believed since the time of Galileo, but the result of frequent, often massive asteroid impacts. And And if the moon had been a target, why not the Earth? Why couldn't a random impact have wiped out the dinosaurs? Although the Alvarez theory is widely ridiculed at first, a small group of scientists begins to find more evidence in the geological strata.
There was shocked quartz grains, which means deformation of quartz in a way that only happens in giant explosions. There were little droplets of melted rock, that is, little spherules of rock that had been melted. There was soot. If you measure around the world and then add up the total amount of soot in this layer, there's enough soot that you have to burn down something like the total amount of forests in the world today, or twice that much, to generate that much soot.
It's hard to conceive just how powerful a blast it must have been, and what devastating effects it had. Imagine the impact of a typical nuclear warhead multiplied 100 million times.
This firestorm came sweeping over this region. A massive, incredible temperatures. Dinosaurs would have been literally fried in their tracks.
As the evidence grows, more scientists are swayed. But many remain skeptical. If such an impact occurred, they ask, then where is the crater?
We had the layer, we had the iridium, we had the hypothesis, but we didn't have the crater. We did not have the smoking gun.
By 1991, geologists think they have it. In an area of Mexico's Yucatan Peninsula, circular, crater-like structures are discovered, buried under thousands of feet of sediment. Core samples taken from the area indicate the age and nature of the material found there, which matches up with other material found all around the world. Analysis shows it all to be 65 million years old. It's hard to imagine any explanation other than the impact of a giant meteorite.
So what are we to make of the tremendous importance of random factors in evolution? Well, for one thing, it tells us that things didn't have to work out the way they did. That, for example, humans didn't have to evolve. If that big meteorite 65 million years ago had missed, we might not be here at all. That should give us a certain humility that our presence here is not a foregone conclusion. I think that's the hardest thing to understand, the hardest thing to appreciate about life, about most of the forces shaping the origins of everything, is that there are steady chemical and physical laws at work every day. They come to work every morning, these chemical laws, these physical laws. And yet the outcomes of them are shaped in really mysterious ways by chance. I think it should give us a lot of pause about what actions we take in the future. Humans are now such an important force on this planet that we have the ability to be a random event, to carry out extinctions, to greatly shape the Earth. And I think we are the first major random event that can actually think about the consequences of its actions. I think we have a lot of responsibility to bring into the next century.
In a universe so affected by chance, we will always be surrounded by mystery. No matter how great our discoveries, there will always be something new. Something we can't decipher. Something unpredictable. Our quest for understanding will always continue. Our science odyssey will never end.
a production of WGBH, Boston.